motto of this particular organization is that your local window to global opportunities in computational biology. The structure of the ISCB Student Council is such that ISCB-SC is an international organization founded in the US in 2006. The Student Council is exclusively composed of students from across the globe, fostering young researchers and early career researchers and their engagement in computational biology. RSG India is regional student group composed exclusively of students from Indian educational institutes under the Student Council. So the ISCB was founded in 2006. It nurtures the next generation of computational biologists with RSGs in over 26 countries. RSG India's purpose is to connect and promote a strong nationwide network of computational biologists, linking local talent to global opportunities. So there are, as mentioned, there are over 30 RSGs active across the globe in over 26 countries, one of them being India. And under the Indian Regional Student Group, we proudly collaborate with premier institutes, including Mahindra University in the Hyderabad, NIT Calicut, and Manipal Academy of Higher Education in Karnataka, as well as the Tamil Nadu Node, which is Sri Ramachandra Institute of Higher Education and Research, which is where we are all from. Here is the RSG India Core Committee. The board is composed of members for the year 2025 to 2026, supported by faculty advisors and visionary leaders across India. Professor Manish Kumar is based in the Department of Biophysics, the University of Delhi South Campus. RSG India has successfully organized impactful events in the past, fostering collaboration, learning, and innovation in computational biology. These are screenshots from the various events conducted in the year 2024 and before. So we look forward to hosting more such events and expect your active participation and contributions. You can reach us through our website or connect with us on social media platforms. I would now like to call on the Tamil Nadu Node Secretary, Syed Abdul Aziz, to introduce the Tamil Nadu Node and the purpose and mission of our team. Syed, over to you.
Good evening, everyone. I hope you all can hear me. Okay. First of all, I'd like to extend a very warm welcome to everyone who joined us on this evening. And I'd like to take a moment to introduce us, the Srihar node of ISCB RSG in Tamil Nadu. We are a student-driven community established in 2025 with a vision of creating a vibrant platform for young and aspiring computational biologists and bioinformaticians. Our goal essentially is to create a platform where bright minds can collaborate with researchers and professionalisms via events, workshops, and webinars such as this one, and encourage learning and collaborations across the field. And then next, I'd like to introduce the team behind the effort, consisting of members from both the undergrad and postgrad level of our bioinformatics department. Here's our wonderful team photo taken at the very first event that we held little to a month ago, actually, held in campus. And a special note and special mention to our advisory board consisting of our faculty, Dr. Dikijam sir, Dr. Abhinan sir, and Dr. Arundhati ma'am. And then now, without further ado, I'd like to pass it on to Abhi sir to introduce our guest speaker. On to you, sir.
Thank you, Abdul. Good evening, everyone. It's a proud privilege for me to introduce the invited speaker of today, Dr. Faraz Shaikh, and it's a great pride for all of us that Faraz Shaikh is one of the alumni of our department. He graduated from the MSc Bioinformatics program in the year 2012 from our own department. Now Faraz Shaikh is the senior scientist at the Oddity Labs in Massachusetts, Cambridge. Um, he has extensive experience in different fields of computational chemistry and computational biology. He initially worked as a junior research fellow at the um, National Facility of Drug Discovery Center, Saurashtra University in Rajkot. Then he went on to pursue his PhD at the prestigious University of Macau. And then during that time, he was also a visiting PhD student at the University of Oxford. And later, he went on to become a postdoctoral researcher at the Institute of Research in Immunology and Cancer at the University of Montreal in Canada. And now he is part of the Oddity Labs, which is in Cambridge, Massachusetts, where he joined as a scientist, and he has grown on to become the lead senior scientist in the same laboratory. So he has been doing extraordinarily well, being a flag bearer of our bioinformatics program, going to different parts of the world. He has even developed a bioinformatics package called as LickTMap, and he's been doing some extraordinary work in the areas of um, high throughput virtual screening, machine learning in bioinformatics. Uh, those are some of the areas where he's expert. So we are all very eager to hear to you, Faraz. Thank you so much for agreeing to give a talk to our students at such a short notice. I communicated to him only like 4 days ago, and here he is. And it's pretty early for him in the morning. It's only 9:15 for him in the morning, I guess. So, and he's a busy father. He has a young family. So, despite all that, he is with us today to share his experience. Thank you so much for us. Thank you so much.
Thank you so much, Abhi sir, to inviting me, and Dicky sir, and Hemlata ma'am, and Arundhati ma'am, everybody who was a part of my master's program. And I think the level of education I got in Chennai really pushed me in my career. And I think you are all who are in SRE right now, they are in a very good place right now for their early career phase. So a good. So my let me share my screen if somebody. So the topic I choose is actually just to give a broad spectrum. Like generally, when we think about computational chemistry, we think a small molecule that gonna bind to the protein, and we generally think of doing docking for small molecules and proteins, and that's where the ending. And I think actually for me also, right, when I was in bachelor's and master's, both in bioinformatics, but more focused on structured bioinformatics, when I was thinking about computational chemistry and drug design, is mostly a small molecule. So, but during my PhD and after the PhD postdoc, where I joined the University of Montreal, I worked with the different modalities like, say, microcycles, DNA encoded libraries, PROTEC, molecular glue. These are all different fields in computational chemistry that are booming. So, and now the other one is antibody design. So it's just like there is enormous scope to us as a scientist to come up with a novel design, novel method, as well as have applications to apply to several different ways. In the beginning, I was trying to include everything that I work on, but I realized that some of the things cannot be shared on YouTube, so I have to remove. Unfortunately, otherwise it could have been very good data to present to the young people to understand like how things can move in a different directions using the computational chemistry. So the motivation of talk was to really understand like there is more than small molecules in the market that really gonna be booming in the coming years, and it's already been booming. Like say like antibody, you say there are 120 or 30 antibody has been FDA approved, and some of them are really comes from the computational chemistry design. So Some of the examples that I'm listing here is like peptide, which are, I think you guys all know peptides. It's been a new modalities other than the small molecules, and there are a lot of tools and are developed to predict the peptide bindings, predict the peptide's properties, create a specific peptide for certain receptors that are very difficult to target with the small molecules. And certain examples is like GPCR, where you have a natural agonist bound into the orthosteric pocket, where you really want to block those natural agonists to bind and activate certain GPCR for certain pharmacology. At that time, you really need a bigger molecules than the small molecules. In some scenarios, that time peptides really help. And I will show you some of the examples related to the peptide. And then there you have something called RNA dock post. In probably 15, 20 years ago, we didn't even think about doing the docking with the RNAs and the DNAs. But now tools are there to come up with this kind of things, which are really helping and booming to understand like how we can use those technology. Other booming field in computational chemistry, which really is the degradation things. Like I'll also show you the examples. There could be two ways of the degradation. It's called molecular glue and the PROTAC. Molecular glue is just bring the two protein, like you say molecular glue. Glue means it's like sticking towards each other, and then create a stabilized complex to lead to the degradations using the ubiquitin pathways. While the PROTAC is like, so one part of the compound binds to the E3 ligase and one other part of the compound binds to your targeted protein that you want to degrade it for certain disease. These are the challenging approaches, because in certain diseases like and some certain undruggable targets that are very difficult to target with the small molecules. These are the new technologies have been come up in the market to really push the drug discovery. And then I just mentioned about the antibody design. So now we also have a machine learning based antibody design. We also have free energy calculation of antibody designs. So understand that some of the topics here is going to be a little bit up above and through our understanding, but I think it's good to reintroduce the new terminology in the early phase of my career, early phase of your career. And I think I believe that now using the chat GPT and AI and Gemini and everything seems to be very achievable and very close to your basket. So that's why I want to not take it a simple approach, but to give you a little bit more complexity about the drug discovery. In the day-to-day life of the computational chemistry, people use docking, do the molecular dynamic, they have a QSAR models, they have free energy methods. We have coming up with a number of other things, like say we in our in-house team in Oddity lab, we modify the algorithm called vSynthesis. vSynthesis is ultra large library screening. Like, so we are screening around 10 billion compounds in the AWS cluster with the thousands of CPUs, and we modify the scripts to run on those things. So now, when we were there in 2007, we used to dock like 10 compounds or 20 compounds. Now docking billions of compounds in just 7 days, or sometimes less than 7 days are also going on. So as a computational chemistry field, the traditional applications are also advancing so fast. And then we also have molecular dynamics simulations. Now the GPUs are so capable to run simulations in a day for 1 microseconds. So then you have a QCR. It is converted to the machine learnings and AI-based models for the activity predictions, solubility predictions, additivity predictions. And you have a free energy methods that are like really accurate in order to understand whether the compounds really binds to the target and how does it correlate to the experimental affinity. And as I explained to you, there are expanded applications that are coming in the like peptide RNA, biology, protein, and molecular glues. And there is a lot of things booming with the AI and ML. Like we have generative designs, we have predictive modelings and automations. So all these goals is to reach out to the target that are previously undruggable, like say KRAS, HRAS, some of the GPCR, some of the complex protein that are not been previously discussed to even have a pocket. So there's a lot of things advancing right now. Me as in a IRIC for almost 3.5 years. Basically, I work for the Bristol Myers Squibb, a BMS company. It's a pharmaceutical company. IRIC is an academic CRO which supports the pharmaceutical projects. So where there I actually come up with a single digit nanomolar for the breast cancer. I cannot present the data, uh, unfortunately. Then I work on the macrocycle. This is another modality. There's like a 10 amino acid, huge macrocycles, and they were struggling to optimize the activity of that macrocycles because the macrocycles were very hard to design. If you have a larger number of amino acids, there is a higher chance of having a flexibility of macrocycles. And that flexibilities and alterable bonds make your life very difficult to predict something. If you induce a minor change, you literally change the whole orientation of the macrocycles. So it is more or less like require a lot of simulations to run with the designs, and also need the expertise to really understand whether the conformation is right to the pocket or not. So we right now this paper is in going on in Journal of Medicinal Chemistry. So it's the novel binding site of KRAS and HRAS, which has not been published before. So it's going to be very good because there are 8 crystal structure we're going to add on to the papers. And some of this modeling has been done by me during that time, which actually improved the affinity to 800 nanomolars. So I think this is a good project. Another project, which again I cannot include, this AT1R. It's a GPCR, which functions for the cardiac and a lot of places that is involved. So here we were trying to design a molecule that have a specific signaling. GPCR is bound to have a different kind of signaling. It could be beta-arrestin, it could be GQ pathways. And when you guys will go into the market for the computational chemistry or drug discovery, I think the GPCR would be much more booming than right now. So it's already been booming, but and then there will be a chance to you to understand like how specificity is really required for certain kind of diseases. And then I also work for something called DNA encoded library design. So this is like a focused DEL library design. DNA encoded library is like you can synthesize like billions of compounds. You have specific E3 ligase compounds, and then you have a DNA linker to take with. You have also attachment point which explain what are the groups that have been added to this, and you have several complex chemistry that runs to generate the multiple, like it could be a two synthesis step or 3 synthesis step to generate the multiple number of compounds for that. And then you also have a screening platform to run all those DEL libraries. So I can also not discuss much about this because again, it's gonna be on YouTube, so it's not useful to give up data on that platform. But what I'm gonna do is I will show off my data, which I already published from my PhD project. I think the Abhisar just mentioned about the LichTMap. LichTMap motives was to design basically to provide a platform to all the organic chemists, those who doesn't understand how to do the docking. So when I was doing my job after my master's, where I joined a DBT project as a junior research fellow, that time I understood like all this organic chemistry desperately need to understand where their compounds really do and what they can bind to, what sort of activity they can have. So I always aim to develop a tools where you can just put your SMILE molecules, SMILE structures. SMILE is a string of information that allows to convert into the 3D structures. So in LichTMap, you just put a SMILE and it will go and identify the target. It will automatically dock to the target. It will generate the binding fingerprints and then compare the binding fingerprint to the crystal ligand binding fingerprints. And then there is a background machine learning model that will predict the PIC50 value for your target. So it's just going to give you a whole table which tells where this ligand binds, how it is comparable to the crystal ligands, and what sort of the activities it can have. So this, and also give you a docking pose and the docking analysis. So this whole platform, the single web server. So web server is right now off, but GitHub is still on. And it's been used like 40, 50,000 times and more usage in USA. So it's a very, very famous tool become in 3 years. Now, the other one that I work in Oxford University, which we come up with the most potent Ebola glycoprotein inhibitor that directly bind in the crystal structures. Its activity is 15 nanomolar. Till today, it's the most potent molecule that has a direct binding proof to the Ebola glycoprotein, because there is no other lab that can do co-crystallization with the small molecule in Ebola glycoprotein other than the University of Oxford. So, and this is another example of virtue screening, how simple virtue screening can lead to the very good project. Other project is the C5AR. It's another GPCR that I was working in collaboration with Pittsburgh University during my PhD. And then later, I deep dive into more onto this whole series to understand where this signaling is going on with these molecules and how the molecules are really working on. Right now, these compounds are very, very promising for me, and that's why I'm not publishing anywhere. And I'm holding for the patent perspective, so we'll see. And just to give you an example about the peptide, because I cannot show my data, but I can show the general data set of how does microcycles really and peptides really helping other people to come up with some of the approaches. So this is a very good example for the GLP-1 receptors. I think GLP-1 everybody knows now. It's a booming target for the obesity purpose. Before it was like for the diabetic, and now it is combined to mostly this Ozempic and all these other things that are booming to make you slim, trim, and a lot of Bollywood people are now using a lot of GLP-1 inhibitors to come up with this thinner and look fit. But this one is not the small molecule, but the peptide. Here they try to come up with novel ways to design the peptide to block the GLP-1 agonist, which they build their own platform. You guys can also look at that platform. This is called Streamline platform. So Streamline platform is like automated design of the peptide by single amino acid mutating and coming up with the peptide that could bind much better ways to the receptors. And these are all machine learning based models. So they generated around like 2,688 peptides. They synthesize and screens and they come up with the lead candidate, which really very selective to the GLP-1, and it has a very good in vitro in vivo data. So please go through that work, which actually tells you like, okay, there are other modalities that are really helping us to come up with the more specificity. And in peptide, it's very challenging to design very specific peptides for certain. This is a new modalities. Here is like you have an undruggable protein that you never been like you don't know how to drug that, because there is very limited information. So for KRAS, HRAS, for certain cancer targets, these are the new modalities. So here what they do is something called molecular glue. Thylamide is one of the examples. This is a very famous one. Molecular glue is, by the name glue, is stick to the two protein and stabilize the complex, and then leads to the ubiquitin pathways for the degradation path. Here you really have to come up with the proper designs to understand how does it gonna bind to this PPI sort of thing, and how it's gonna be stabilizing that. So there are a lot of computational ways to do this, and there is other way called PROTEC. PROTEC is one part of the molecules binds to the E3 ligase, and the other part of the molecules binds to your target protein that you want to degrade it. So both has a binding site and binding pocket. So what does it really do is that you have to come up with this proper linker that give a proper confirmation of both side of protein part of to bind to the exactly to the pocket. So these linker designs are been a lot of AIs and computational ways to come up so that it gives proper confirmations of both part of the binders. So this is also a new technique that computational people are doing right now. And there are a lot of virtual libraries to come up for the PROTEC and molecular glue. So if you guys are interested, also go and look at all these things, which is really booming right now. So let's move into my own project, and I will give a little bit rush now. So this is the work that I did in on University of Oxford. I identified the molecules for the most potent molecule for the protein. When I joined their group, they had already published work where the turmifin, one of the cancer drug, actually binds to the pocket that I am the arrow down here. And my role was to identify something in 4 months to block this binding site with the novel molecules. And then we had understanding how does this tormethine actually binds to the pocket, but the pocket was huge. It's like it could fit more than one molecule so into the pocket. So identifying something for that kind of pocket is getting very challenging, because you can have a lot of conformation in the same pocket with a different kind of compounds. So the first thing what that they published in paper Nature, that they understood, okay, the tormethine can destabilize the whole complex and they could bind to a specific protein. So what I did is I took the non-binders first and tried to dock with the Schrodinger tools, with the Glide models and QPLD, QMM based docking, and try to correlate with the experimental affinity that they had to see what should be the good scoring models that we can use to come up with the screening filtering pipeline. We had so when they say, okay, you have 4 months, so 4 months you basically want to have a natural compounds or something that are derived from the natural compound, not very toxic or synthetically and synthetically feasible. All these things you have to be in your mind. So I had this library from Zinc database. Basically, it's downloaded from a traditional Chinese medicine library. It's 2.5 million compound, and then I docked to the high throughput versus screening to got around 416 compound, and from there, Schrodinger has a two kind of tools, which is like a standard precision method for the docking and an extra precision for the docking, and there is also something called QMMM based docking. So this is more detailed stuff, but what I want to tell you that we end up with the 16 compounds, and out of 16 compounds, we brought 8 compounds. From these 8 compounds, we actually found 2 compounds that are doing very good. This is the list that we actually brought, and we found that there are 2 compounds which very look similar, but one of them is actually almost 50 nanomolars to the destabilization soluble glycoprotein, and it also has a direct binding evidence. And then we co-crystallized this with the Ebola glycoprotein and published in JAMET-Chem. So it's a good short project that we did. The other one is the active learning based C5AR molecule identifications, as I mentioned. So the C5AR is a GPCR for the inflammations and other cancer diseases. There is already a known drug that is in the market that's called avacopan that binds to the orthosteric pocket of the GPCR. So what my goal was to find something that is competitive as to the avacopan, and basically finding was the antagonist. But actually, this is unfortunate discovery that we end up into finding the positive allosteric modulator. So the difference between positive allosteric modulator and antagonist is antagonist block the GPCR signaling, while the positive allosteric modulator is actually enhance the GPCR signaling in the presence or absence of the natural agonist. So we try to come up with the method. We use a lot of active learning based Schrodinger active learning based technique to come up with some of the molecules. We did the functional assay using the team of Pittsburgh University, and then further explore the mechanism actions from the IRIC GPCR pharmacology famous guy Mr. Bowyer team. So how does we actually come up to the specific proteins to understand where this really works? So if you look at this is a TSNA plot. We run the screening, we run the docking, we got a lot of hits, and we try to see which are the closest hits to the known ligands of CPAR. So if you look at the red dots, these are the known ligand for CPAR. These are the green is the enemy in space, and the blue is the zinc database space. So here we have some of the blues that are closest to the known inhibitors in terms of the ligand similarity, and there are the green cluster which is very close to this type of known inhibitor. So this green cluster is become to our own positive allosteric modulator whole series. So it's very similar towards the avacopan and the other known binders, and also does the same impact to the C5AR. So when we first screened the Pittsburgh University, some of the compounds, we see that some of the compounds are really doing the reverse. What we actually wanted them to do, the antagonist, but it's actually doing the agonist part of it. And when we aid the natural C5A, that is a natural peptide that binds to the C5AR, it's actually boosts, it gives a synergetic effect to increase the activations. So it's actually working less than a positive allosteric modulator. And then we come up with the other compounds of the same derivatives and see the whole series is actually doing the same thing. So it's actually a novel molecule for that particular chemokine receptors for that particular purpose. Now I'm into the patent part of this whole series to be a PAM molecules for that receptors. PAMs are not generally desirable for this target, but some of the cases is really helpful to have a PAM for even for the C5AR. And then we try to come up with a novel assay where we try to understand whether this compound really binds and blocks to the avacopan binding site, because the docking was actually done on the avacopan binding site. So we come up with the competitive binding assays. I think Dicky sir can explain you much more about the assays, but here is to tell that if we increase the concentration of my compound, the PAM series, we see a antagonist effect going down of the avacopan. So that tells you, okay, so it replaced the binding of the avacopan, one reason, one hypothesis, or it is binding somewhere that influenced the binding of avacopan. So, but at least it helps to understand, okay, so there is something that these compounds are doing that that even the inhibitors can lose the activity. The other work that I mentioned about the Lictimab, so we had a 17 different class. I wanted to cover as many as we can so that people can have activity prediction of their own small molecules. So what we do, we have a small molecules. We generated a combination of fingerprints, Morgan, mass, and daylight, and this will identify a similar ligand from the PDB bind data set, which is closest to your ligand, and then those ligands that are close to your ligand will have a target already bound in PDB. So your ligand will go and binds to the PDB and then automatically dock in the PS arena and generate the binding interaction fingerprints and compare the binding interaction fingerprint with your crystal ligand. Please read the paper for the more detail. I think it's going to be very interesting for people who really wants to computational chemistry. These are the known target prediction servers in the market. None of them can actually automatically dock your compound. None of them can actually automatically predict the activity of the compound. So this way, Lictimab is really special. It has the database from PDB-Bind 2017. Now the PDB-Bind 2025 is already upgraded. There are also a target class that I mentioned. I basically put kinases, ligases, helicase, and the disease based like HIV, HCV, and tuberculosis. A lot of things I made to. These are the target class. We have a total 2008 PDBs from the kinases. And we also had a lot of machine learning models that we tried to come up with this for the activity prediction, because all this 2008 compound had the activity. So we take the small molecules and their activities on, and then we create a random forest model for all this class and try to build the models and put it in the background to explain the activity. And what really especially we do is called benchmark set. We actually brought all the GEM-ATChem molecule recently published in 2018, which are not in our training set, to see whether our models can perform better to the diverse set of the ligands. So just to see if we can have a better applicability domain compared to the other servers. And generally, people just have a test and train split, and in that test split also we have something called nested cross validation. You have some part of the test set, also some part of that will go into cross validation. So total we had a 98 molecules from the Journal of Medicinal Chemistry. The reason to choose Journal of Medicinal Chemistry is that they are very accurate in terms of the data. So we try to first get into understand what are the different fingerprints that we can use. So like we have used several, like 8 or 9 fingerprints from RD kits. So it could be a Morgan, mass, daylight, topical, pharmacophore based fingerprints. and then come up with a combination that works much more better for the PDB-bind data set. So these are the 6 different fingerprints in individually how they perform to identify top one ligand, targeting top 1, top 5, or top 10. But when we combine them to Morgan, Mass, and Daylight, we see the performance and boosting. So we take these final combinations in our web servers. We had our own tools for the docking called PSOVINA 2.0. It works on the swarm-based optimization method, and it just merged with the target tool so that your compound can automatically dock in the background. Something called binding interaction fingerprints. So once your compound dock, it will generate the interaction fingerprints. Interaction fingerprints will like have a binary string which denotes what sort of interactions are. It records what sort of interaction crystal ligand has, and then it try to compare with your own known ligands and see if we have the same conserved interaction as to the crystal ligand. And then you do a Tanimoto score to compare the scoring. About the Lictimab class specific experiment, what we really find is that each class will have a different sort of accuracy because different data set that we are bringing. Some data IC50s are cell based, some of them are from the binding assays. So it's a lot of differences. So if you look at the beta secret test, we always perform like almost 97% time highly accurate, and then followed by bromodomains or carbon anhydrase or the ligase or the HIV. HIV is also very much like a structure-based data set, so that's why the benchmark you will see the very high performance. Where in kinase is a lot of diversity and the compounds could come from the different experiments, so that's why the scoring, the performance was not very good, but combination of MMD still perform well. So more details are in Journal of Bioinformatics papers. It's an open source. As I mentioned, we took all the 5 target and we tried to compare with the existing servers also, like Swiss target, C, SuperPrep, and we're seeing the Lictimab is overall performing very well with all the targets. And some of them we really literally outperform the HitPick, SuperPrep, and C. We are very close to the Swiss target predictions. And in top 5, some of them we are away from. And these targets are from the GEMATChem paper, so not even included in Swiss target, not even included in Lictimab. So it's very unbiased comparison. And then we had machine learning models that we built using the random forest algorithm, which is nested close validation part of it. So we try to nested close validation generally used to minimize the error in your models as much as you can, because the data set is limited and we always wanted to work on the true specific and clean data set. And any machine learning models, if your data sets are not right, you're not going to get anything better in terms of the predictions. So we use this method, but before that we used several other tools, several other methods to come up with these ideas. And not very surprising, HIV is doing very well because HIV is all the molecules that you see in the market, they are structure-based design. So, and they correlate very well with the score because they have a PKD value. PKD means the direct binding affinity values, and the where you have a direct binding affinity values correlating with the binding scores and the fingerprints is really going to push your models. It's very true positive data set. And then you have what's performing is the estrogen receptors, and you can understand why, because estrogen is like, it's a decade-old receptors, and many people have published a lot of things about the estrogens, and many of the molecules will have a lot of false positive data set into that particular things. But still, it's fine to have that kind of predictions for the early understanding, just that how your molecule is going to be working. Very good example of case study from HIV papers. That paper was published in SCS MedChem. They come up with a novel design of these molecules to see how and improve the activity of the molecules. So what we did is we just put our compound into those compounds into the Lictimab. It identify almost all the time to the same target and also predict the activity. And the correlation was very, very good. It's almost like 0.83. And there is another machine learning based HIV prediction tools, which is HIV portal, and it's almost like 0.56. So the model that we built with a true positive data sets is really outperforming the existing method. So it's that's why This is 3 years old data. I'm putting up that it's been like 35,000 views, 7,000 files have been uploaded, and people have crashed this Lictimap because it was open source and free. People were using like a day in hour. Most of the users from USA, and surprisingly, second is not surprisingly India and then China, but like a lot of usage to these tools, and then the server crashed. So we only have a GitHub now. Other publication that I do during my